we shall be discussing about neurulation basically. So our point of concern today, by the end of this discussion, you must be able to define what neurulation is and be able to tell the basic processes that occur during neurulation and then the clinical presentation to you as a medical practitioner in day-to-day setting. Those are our take-home for today's discussion, and by the end of this, we should be able to tell those things accurately. So what do we mean when we talk about neurulation itself? Basically, when we are talking about neurulation, we are talking about formation of neurotube. And for you to appreciate this concept very well, you must be able to do a review on our discussion on gastrulation and notochord formation, because that is what we are actually going to be building on this very day. So why is it important to know this basic procedure or these basic two procedures that I've told you? I told you it's important for you to know gastrula or gastrulation, which is the formation of trilaminar disc or 3 germ cells, germ layer from a 2 germ layer, as you can see on our image today. And then be able to tell or know about notochord formation. Because normally, after formation of 3 germ layer, which is called the trilaminar disc, as you can see, during gastrulation, we form a disc with 3 layers, which is the uppermost disc. We call it the ectoderm or ectodermal layer. And then the middle layer, which is the mesodermal layer. And then we have this other layer, the innermost, which is the endodermal layer. Now, during notochord formation or the formation of notochord, we actually form a notochord which occurs at the center, which is normally at the mesodermal layer of the 3 germ layer. Why is it important to know about notochord formation for you to appreciate neurulation? It's because the notochord acts as the primary inducer of neurulation. The notochord has 3 basic roles or function in this particular discussion. One, it's the primary inducer of neurulation. That is notochord. Our notochord is the primary inducer of neurulation. Of course, it has other 3 more functions that I can mention just, but this is the most important one that you must be able to identify. The other one is it acts as the central axis of the developing embryo. This is our notochord here. It demarcates the center and it runs through. So it's the central axis, and also it forms the center which the vertebrae, the vertebral bones and the cartilages are formed around during development. So after appreciating those 2 major processes, which is gastrulation and notochord formation, you will be able to now appreciate our main topic of discussion today, which is neurulation. So how does this neurulation occur? After we have formed the 3 germ layer, we have the notochord at the center, and then we want to form our neurotube. So how does this neurotube actually develop? So we start from the notochord still, because I told you it's important to appreciate the formation of notochord. If you review our other videos on notochord formation, what happens is the notochord begins to secrete growth factors. And these growth factors go up to the ectodermal cells, which actually is overlying the notochord. What happens to these cells which overlie the notochord? It undergoes differentiation. And when they undergo differentiation, they form what we call neuroectodermal cells. As these growth factors are being released, the cells which are overlying the notochord differentiate and form what we call neuroectodermal cells. These neuroectodermal cells also undergo further differentiation as more and more growth factors. These are growth factors going up, growth factors. As more and more growth factors are being released by this this is notochord. As more growth factors are being released by the notochord, these neuroectodermal cells begins to thicken. They thicken in the area which is above. So this is the neuroectodermal region. These cells above begins to thicken and thicken as they proliferate. They thicken and thicken and thicken. So this thick area, which is overlying the notochord, becomes what we call the neural plate. The neural plate. So neural plate is basically a layer of ectodermal cells which has undergone proliferation and thickening. And that area is specifically overlying the notochord. So what happens? These cells which are in the neural plate begins to undergo what we call differential growth. The cells at the margin of the neural plate grow faster or begin to develop faster, mitotically dividing faster in comparison to those other cells in the neural plates which are at the center. That is one factor that is going to happen. The second thing, this mesoderm that we talked about here initially, this mesoderm, the area which is nearer to the notochord also proliferate and thicken. So it pushes the margins of the neural plate up, pushing it upwards, causing elevation of the margins of the neural plate. So this is what will happen. As this is our notochord, as I said already, secreting what? Growth factors. And these growth factors cause the cells which is overlying the notochord to proliferate to form neuroectodermal cells, which thicken to form what we call the neural plate. So the neural plate, the margins of the neural plate begins to elevate and elevate. Now, as it's elevating or rising further, it forms what we call folds. And this is what we call the neural folds. It forms the neural folds. Now, as the elevation continues with mitotic cell division, the center, which was growing lesser and is thickened, it involutes. In other words, it sinks or it goes downwards toward the mesodermal layer. And this center, which has involuted, it forms what we call the neural groove. So there is a groove and then the neural fold that has been formed. So in this area, we have what we call the neural fold here, the neural fold. Then at the center, we have what we call the neural groove. What happens next? The cells normally of the neural folds begins to grow towards the midline. They grow towards the midline. This side also grow towards the midline. Grow towards the midline. As they grow toward the midline, they eventually fuse. And as they fuse, they finally form what we call a tube. But important to note is that the cells which are at the tip of the neural fold normally don't participate in the fusion. These cells actually detach and fall into the mesodermal area, and then they form what we call the neural crest cell. So if you come to this other diagram, you'll find that the folding has been completed, and then we have what we call the neural tube has been formed. Then the cells, of course, our notochord will be below here. Then the cells which were at the tip will also come aside, and this is what we call the neural crest cells. The neural crest cells. So another thing that I want to bring to your attention is that as this neurulation occurs, some cells, or the neural crest cells, actually remain at the ectodermal layer of this previous trilaminar disc that we have formed. So now the cells, or the neural crest cells, that actually remain here, it's what we call the ectodermal placodes. Ectodermal placodes. Placodes, it's like this. Ectodermal placodes. So it's important now that we have known that we have formed the neural tube, we have the neural crest cells, and we have the ectodermal placodes. Why is it essential for us to know this? Because during the process we have been talking about neurulation, all the way from where we had the trilaminar disc, then we have our notochord, and then there is elevation of the margins of the ectodermal or the neural plates. There is active involvement of cell division. As we are having cell division taking place actively, there is increased duplication of DNA. The DNA has to be duplicated so that each cell that is being formed has equal number of chromosomes as the parent cells. During this process of duplication of DNA, there is need of folic acid. So this is the process where we need a lot of folic acid during the process of neurulation. That's why you'll find in the hospitals, the doctors recommend folic supplement in all the mothers who are actually in their prenatal or in their early days of gestation. Recommended saying 3 to 5 milligrams of folic acid is utilized per day during this process of neurulation. So what is the fate of these other cells that we have formed? We have formed neural tube at the end of it all. So what happens to neural tube? This neural tube actually has an opening in both areas. You have seen that we have formed just a folding. So it's actually like this. So we have what we call neural tube. So this tube has an opening on the cranial side and an opening on the caudal side. This opening on the cranial side is actually called the cranial-neuropore. And then the one in the caudal side is called caudal-neuropore. Caudal-neuropore. And all these pores actually have to close because the cranial side of the neural tube, in future it enlarges and actually forms our brain. And then the caudal side remains as a part of the neural tube, which will actually form the spinal cord. Failure for complete neurulation or closure of these neuropores will form what we call neural tube defect. Neural tube defect. So what would contribute to failure of this closure? It may be as a result of maybe folic acid deficiency. It might also be as a result of genetic factors, because the genes actually play a very essential roles in this particular aspect of neurulation that I've talked about. So what happens to our neural crest cells that we have formed? The neural crest cells are also very, very, very essential. The neural crest cells will also further differentiate to form what we call the melanocytes. Some will form the Schwann cells, others will form or will migrate to form the cells of the adrenal medulla, others will form the cells of the posterior ganglia of the cranial nerves, and then others will form the cells of the ganglia of the autonomic nervous system. The same way, the cells in the ectodermal placodes, others will form basically the cells that participate in the formation of the peripheral nervous system. Okay, so we have seen that by the end of neurulation, we have come up with actually three things. This is neurulation. We have come up with the neural tube itself. The second thing, we came up with the neural crest cells. And then the third thing that we came up with is ectodermal placoids. So these are the three aspects that we resulted with at the end of neurulation. So what happens to each of these? The neural tube itself is actually our future central nervous system. So it will form the brain and the spinal cord actually. Then the neural crest cells will further differentiate into a number of cells actually. One, it will differentiate to form the melanocytes. It differentiates to form the Schwann cells. It differentiates to form the cells of the autonomic ganglia. It also forms the cells of ganglia of cranial nerves, and then finally it forms the cells that form the posterior root ganglia. That is actually the fate of the neural crest cells. Then what happens to the ectodermal placoids? The ectodermal placoids actually will result in the formation of basically three things: the sensory root ganglia, this one we shall look at it later, the hypophysis, and then finally the inner ear. Thank you for attending. It has been nice having you. Have a blessed day, blessed evening, and blessed night.